Hello, everyone. My name is Liliane Conteville. I am a postdoctoral fellow at Embrapa Southeast Livestock from Brazil, and my work is entitled Taxonomic and Functional Diversity of Rumen and Fecal Microbiomes in Nelore cattle, linked to diet and production phenotypes. The main interest in this is because Brazil is one of the largest producers of beef in the world, and more than 90% of the Brazilian beef cattle herds are composed of pure and crossbred Nelore. And Nelore, like other ruminants, harbors a complex microbial community in their gastrointestinal tract, which plays significant roles in the cattle's health and physiology. But despite its importance to the Brazilian economy and human feeding, few studies have characterized Nelore microbiomes. So here we used shotgun metagenomics to explore the microbiomes of Nelore that received two different diets and that differed in their feed efficiency and methane emission phenotypes. So we had two Nelore groups. The first received a conventional diet consisting mainly of corn and soybeans, while the second received a byproduct diet that also consisted of corn, but in addition, it had citrus pulp and peanut shells. We collected samples from the ruminal content and feces of these animals and generated metagenomes that were filtered with Trigonometric and then mapped to a bovine genome using Bowtie2. Taxonomic and functional classification were performed by Kraken2 and Superfocus, and Lefts and Maslin were used to identify features associated with the dietary groups and phenotypes. The indices used were residual feed intake, RFI, and residual methane emission, RME. First, we observed that the ruminal microbiome showed higher microbial diversity than the fecal microbiome, and their composition was clearly different. We can see here the segregation among the metagenomes from the two environments. Their potential functions were also different. For example, the ruminal microbiome had a higher abundance of functions related to monosaccharides and the degradation of proteins, while the fecal microbiome had a higher abundance of functions related to D-oligosaccharides and proteins biosynthesis. Then we explored the differences between the dietary groups. At the phylum level, we see simpler differences, but considering the genera, we identified 89 bacterial and archaeal taxa that were differentially abundant between these groups. In the functional analysis, we identified some metabolic adaptations that occurred in response to the different dietary compositions. For example, in the conventional group, there was an enrichment of functions related to maltose and maltodextrin utilization, which are produced when starch is hydrolyzed. And in fact, this diet has a considerably higher amount of starch. And in contrast, in the byproducts group, there were enrichments of functions related to fructooligosaccharides and xylose, which both are known to be present in fruits. And this diet had citrus pulp in its composition. And we could find many other associations like those. And even considering the phenotypes, we could see the effect of diet on the microbiomes, since only the efficient animals fed the conventional diet and the low methane emitters fed the byproducts diet had a higher microbial diversity in their fecal microbiome. But besides that, we could associate multiple genera from rumen and feces with the increase or decrease of the phenotypes indices. For the RFI, we generated a PCA with the functional profile of the taxa associated with RFI, and we observed segregation into 3 clusters, showing that there are differences among the functional potential of ruminal and fecal taxa associated with inefficient or efficient cattle. One important difference is the ruminal taxa associated with efficient cattle having more functions related to carbohydrate metabolism. In the functional profile of the taxa associated with methane emission, we identified enzymes involved in the production or consumption of hydrogen and formate, which are important intermediates in methanogenesis, and also observed the presence of coenzymes and cofactors essential for the activity and function of methanogens and methane production. So these results have the potential to contribute to the development and implementation of strategies to reduce the environmental impact of livestock and also improve the animal production. I'd like to thank for this opportunity of presenting my work. And if anyone have any questions, you can send me an email to lilianeconteville@gmail.com. Thank you.